Hey friends, I try to make a video once a week, and this week I'm in Boulder, Colorado visiting my granddaughter. And my granddaughter unfortunately is off at the doctor's regular pediatrician's visit to check up on her. She's looking good, so I'm not going to talk about her today. Anyway, here I am in this cheap hotel room drinking cheap hotel coffee, feeling obligated to say a few things on YouTube, and what I thought I'd do is address a question I got in my email. So exciting, right? Let me show you, and I will read the question. The question is homologous structures with different developmental pathways, and my correspondent says, I know you've been asked that question before, but in my opinion, you didn't reply in a proper, detailed way, sir. Well, okay, I guess I better answer in a proper, detailed way today. Yes, how can there be a homologous structure with different developmental pathways and maybe different genes? Again, to any developmental biologist, they'll say, that, well, that's no surprise. That's kind of what we expect. And then he says, according to this article by JW, I'm apparently supposed to know who JW is, the ID advocate, and they make a problem for evolution. And he cites an article in Access Research Network, ARN. Okay, I can tell already it's going to be a bad article. And he says, I hope you reply if you have time, Prof. Thanks. I don't know quite what to make of this question. It sounds a little bit trollish. This is the kind of thing a creationist might write to me thinking, aha, I finally got an evolutionist trapped. And here is my official formal citation that blows evolution out of the water. Unfortunately, it's on the ARN network site. So no, it's not going to do that. Okay, so I've got the article here. I printed it out. It's 7 pages long. It's 7 pages of crap. Okay, first thing I notice is the title is Homology, a Concept in Crisis. This is the most facile, superficial way to approach it, because this is apparently the work of these authors who went out and asked what problems are scientists having in evolution. And I can tell you, as a matter of fact, you can find tons of articles, real research articles, that mention the problem of homology in their titles. And you can tell how excited they were to find this, because it means, oh man, there's a difficulty, therefore evolution must be false. So that's the first bad impression. I mean, seriously, I have given undergraduate students term paper assignments to write about the problem of homology. It's that common. It's that well recognized that even undergraduates are exposed to it. Okay, second problem, the authors. The authors are Jonathan Wells and Paul Nelson. 2 intelligent design creationist hacks. They're terrible. You know, on YouTube you'll find a lot of people having debates and arguments with people like Kent Hovind or with Ken Ham's ideas. Ken Ham does not personally debate anyone unless you're Bill Nye. And you'll run into that all the time, and those 2 guys are ignoramuses. They know absolutely nothing about evolutionary biology. So they're kind of an easy target. Jonathan Wells and Paul Nelson are a little different in that they have a little bit of knowledge, just a little bit, enough to be dangerous. So they are familiar with the academic literature. They get out and they read this stuff, and then they mangle it. And they make it look like, hey, we're actually doing serious academic scholarship here. We follow the forms. We use the language of scientists. Therefore, you're supposed to take us, you know, as credible sources when they're really not. They're terrible sources. So, right away I'm having problems with this article. Okay, but let's look at the abstract for the article. It says, before Darwin, homology was defined morphologically and explained by reference to ideal archetypes. That is, to intelligent design. I'm having a problem with the first sentence in the abstract, because what they continually do is they confuse, process with outcome. That, yes, they define things in terms of ideal archetypes in the 19th century, for instance, but that does not mean that they had any idea of the process that produced them. So you can't exactly say that this was a good defense of intelligent design. It was intelligent design by default because nobody had thought of anything else at the time. Okay, then it goes on. Darwin reformulated biology in naturalistic rather than teleological terms and explained homology as a result of descent with modification from a common ancestor. Okay. Descent with modification, however, renders design unnecessary only if it is due entirely to naturalistic mechanisms. Again, I'm sort of in a place where I have to say, well, sure, I can sort of agree with that. That if you can come up with an unnatural mechanism, a supernatural mechanism that produces design, then I would have to accept it. Show me the evidence. They're not going to show us any evidence for that. As usual, their argument is going to rely on saying, well, here's some natural mechanisms and they have problems, therefore we have to accept intelligent design, which is bogus logic. It doesn't work. Okay, so he goes on. 2 such mechanisms have been proposed. Wait a minute, only 2? How deeply did they get into the literature here? Anyway, 2 such mechanisms have been proposed: genetic programs and developmental pathways. This is the kind of thing they're always pulling, and it just stumps me because sure, those are a couple of buzzwords, and they're pretty commonly used in the Evo-Devo literature, but you haven't addressed the concepts yet. And he goes on to say, but neither one fits the evidence. This is going to be a non-stop head scratcher for me, because this is going to go on and on that, yeah, that neither one single concept fits all of the evidence. That evolution is a complex process. There are multiple design mechanisms. And notice I use the word design mechanisms because that's the way they talk. There are multiple mechanisms that generate these appearance of design. That does not imply, however, that they are design. Also, what we're going to see as we look into the literature is that there's going to be a lot of different ideas about how these things happen. And simply saying that one doesn't work and this other one doesn't work in this other condition, therefore the whole thing is wrong, is not good science. And then the clincher here. Without an empirically demonstrated naturalistic mechanism to account for homology, design remains a possibility, which can only be excluded on the basis of questionable philosophical assumptions. I would turn that right around on them. Without an empirically demonstrated naturalistic mechanism. We have empirically demonstrated naturalistic mechanisms, multiple mechanisms, and the intelligent design creationists have demonstrated none. Therefore, we should throw them out, not us. Okay, so the rest of the article is just noise. But let me just jump ahead to one other little problem here. And this kind of drives me nuts too. I'm going to the last pages here where they've got a long list of references. And they do reference these people throughout the paper. I mean, for instance, I see here Perry Albrecht, Sidney Brenner, Eric Davidson, Scott Gilbert, Brian Hall, Richard Lewontin, Fred Nijhout, Rudy Raff. They're all in here. Neil Shubin. This is something else they do that is dishonest and disreputable. So throughout this paper, they're going to cite these guys. These are all really well-known evolutionary biologists and Evo-Devo people. They have intentionally sought out some of the best known names in the field and patched them together in their essay. And here's the thing. All of these scientists recognize the problem of homology. Really, we've been talking about this for decades, for a century or more, that there's a problem with interpreting homology in different organisms. And then they're going to turn around and conclude that, well, then evolution has got a problem. We're going to have to throw out naturalistic evolution and rely instead on intelligent design. Again, they have no evidence for this, but that's their basic argument. But the thing is, what they have to do is they have to cite little tiny bits of these scientists' papers where they bring up problems in interpreting homology. And then they make this grand pronouncement that it's an anti-evolutionary thesis. If a student did this, I would flunk them on the spot. Because what you have to do to make that argument is you have to read fragments of these authors' papers where they discuss seriously and in depth problems in interpreting evolutionary evidence. And then come to a conclusion that's exactly the opposite of the things the authors do. That is profoundly dishonest. It would say that they haven't read the whole of the paper for its meaning, that they're just extracting little bits, that what they're doing is literally quote mining. So this is a great big exercise in quote mining. Again, that's not credible scholarship. It ought to be thrown out on that basis. Okay, but let's talk about their actual argument. So when they say that there's two proposed mechanisms, that's not quite right. When they try and narrow it down, what they basically do is they cite a couple of different disparate problems in interpreting homology, and then mush them together and say, well, the whole thing's wrong. So, for example, one of the things they point out, and again, this is true. This is a fragment of real science, that when you look at different organisms and different genes, what you find is that sometimes the same gene in different organisms generates different structures. So they use as an example PAX6. PAX6 is a master control gene. It is a regulator of development, and the expression of PAX6 induces eyes. In flies, PAX6 induces fly eyes, big red compound eyes. In mice, it induces camera eyes like we have. And you say, how can this be? You're using the same gene, but you're getting different outcomes. This is a problem for evolution. They say, well, no, it's not, actually. Because what it is it reflects something I've mentioned before in my videos, is that there are a lot of genes that work together to generate complex structures in an organism. It's not like there is one gene that says make an eye, and then an eye appears. It's one gene that says, okay, switch on these other genes, and those other genes start a cascade, and that cascade leads to expression of other genes that leads to an eventual outcome. There's 1,000 steps along the path from that initial gene, that initial pattern of gene expression, to the final output of an eye. And all of those are subject to change in evolution. So let me emphasize that. And that is that there isn't a discrete teleological function for a specific gene, or there are multiple parts that all work together to create a structure. And when we talk about the eye being homologous, we're saying that the bits and pieces have different histories, they have different relationships, but we can trace those similarities between a fly and a human being. It doesn't mean the whole thing is exactly the same. It means that there are pieces that have operated in evolution, that have been shaped by evolution, that have changed over time, but we can trace them back to a common origin. So that's one problem they have. Excuse me, I have to drink some more bad hotel coffee. Okay, so that's one problem. That the same gene can produce different outcomes in different organisms. And they're kind of freaking out about that. Evolutionary biologists aren't. It's kind of what we expect. The other problem they have is that you can have the same outcomes produced by different genes. What does that mean? That means that over evolutionary time, things have changed around so much that there are deep genes early in the pathways that have changed between different organisms. Okay, and you might say, well, that's a problem for homology. We make the same thing, but we do it in different ways. And as a biologist, I have to say, well, no, that's not a surprise, because what we find is a great diversity of different ways that things happen in evolution. What we could talk about here is, for instance, convergence, that a desired outcome may be produced by different gene functions converging to produce similar outcomes. We also talk about things like parallelism. So, if you get some fundamental detail, some feature that's really important in the organism's life history, you may expect that there is selection for greater robustness in its development. So what do we find when we look at this stuff? We find that there's a whole bunch of genes that contribute to the pathway that leads to the outcome. And then what you can have happen in evolution is evolution in one lineage can pare away maybe one set of parallel pathways, and a different organism, a different set of parallel pathways get shaved away. So when you look later, you find they're producing the same thing, but they're using different aspects of an ancient program. So again, that's not surprising at all. It's exactly what we expect to see happen. Wells and Nelson seem to find that problematic. So let me give you a couple of specific examples. In evolution, what we find is that there are many different pathways that work in very similar ways, and all pathways are subject to selection. They are also subject to random drift and all kinds of change. So in the 19th century, there was this initial idea, before they had actually discovered genes, that maybe the reason for the archetypes we see is that all change is hierarchical. That is, that in primitive organisms you have one set of pathways, and that you make a new kind of organism by stacking another set of pathways on top of that, and another set on top of that, and another set on top of that, so that you can go back to the original pathways. And what you find is those are a foundation that are locked in. You can't change those without the entire edifice tumbling down. So there's the idea that, okay, these are all rigidly determined. How could evolution possibly change those? That's a very Hegelian sort of way of looking at things. Is it true? No, it's not true. It turns out that as we acquired knowledge about genes, and we actually looked at the genetic pathways that are involved in these structures, we found that they're subject to change at every level of development, even the earliest genes. And this should have been obvious, right? Because we can just look at ourselves and see an obvious example. We have little embryos, right? And human embryos, mammalian embryos in general, are going to rely on implantation in a uterus to get their nutrients. So practically the first thing that developing embryo, that little morula, has to do is attach to the uterine wall. It has to develop a set of external cells that provide these external membranes that link up to the mother's uterus and facilitate the exchange of all kinds of nutrients and information. That happens early. It happens in mammals. It doesn't happen in reptiles, right? It doesn't exactly happen in marsupials even. So what we've got is an evolutionary novelty in the development of the mammalian embryo that occurs in the earliest stages of its development. So you know that, you should be thinking, yeah, it's kind of obvious that things can change all over the place. And there's no foundation that is fixed. When people talk about the problem of homology, this is what we mean by the problem of homology, that it's not so trivial as to be able to say, well, this set of genes are fixed in their function for all time, and therefore we can relate them to one another. Because new patterns arise all the time. That's what evolution is all about. It's about this constant turmoil, this constant change. It produces new variations. All right, let's think about another example. A lot of our knowledge about early Evo-Devo, the stuff that initially started Evo-Devo, was work done on Drosophila, so the fruit fly. Fruit flies are weird. Fruit flies are highly, the Hyrdiptera in general are extraordinarily derived organisms. They're very different from other insects even. And so we've got this very complex pattern that arises those. That's what we understood first. And then we started looking at other organisms, other insects, and other organisms like people, and we started seeing variation from that. And we're not shocked by that, because what we've seen is a consistent pattern in these organisms. So, for example, one of the earliest genes that is expressed in Drosophila that defines anterior-posterior patterning is a gene called bicoid. Bicoid is basically the master generator of the anterior-posterior axis of the animal. That if you knock out bicoid, you don't form a head. If you express bicoid inappropriately in the wrong place, you can get flies with two heads. So it's really controlling that part of the pattern. Yet, what you find is when you look at other insects, it doesn't do that. Bicoid isn't even a big player in axis formation in other insects. It is not at all a big player in humans. We don't even have, strictly speaking, the bicoid gene. When we look at in other organisms, what we find is that there is a HOX3 gene that is homologous by sequence with bicoid. So bicoid is a HOX3 gene that got plucked out and recruited to play this role of setting up a gradient that forms the axis of the animal. Other insects, nope, they don't do that. If we look in vertebrates, for instance, have a gene called goosecoid. So goosecoid is basically, it's the product of a fusion between a bicoid-like gene, or HOX3, and another gene in flies called gooseberry. And it plays an important role in defining dorsal ventral patterning in the early embryo. And then here's our problem of homology again. What is the homology here? If goosecoid is a combined version, a fused version of two genes in the fly, and is defining a different axis, is it homologous or not? I mean, we have a fully naturalistic explanation for how it arose. The fact that the homology is complicated doesn't mean that we reject the whole idea of evolutionary change. So this is the kind of game that they're playing throughout here. And lots of times we actually have good explanations for some of the changes that occur. So to throw another example at you, yeah, I'm full of examples. Segmentation in insects. Again, we figured this out first in flies, and flies are bizarre, strange little creatures that have been highly shaped by divergent evolution, so they're kind of complicated to relate. But when you look at insects in general, what you can find is, for instance, there are two categories of insects. There are short germ band and long germ band insects. The germ band just refers to the body axis that forms early on in the embryo. In short germ band insects, you got this mass of embryo, and then what happens in development is, if I imagine this is my embryo, it will form one segment, then a second segment, then a third segment, then a fourth segment, and more segments. It does it sequentially over time, and it's actually driven by what's called a Semitic clock. There's a ticking clock, molecular clock, that generates this pattern. That's the same thing that we see in us vertebrates. We have a Semitic clock that generates this pattern in a progressive way in our development. So that's what a short-germ band insect is. A long-germ band insect, on the other hand, has the whole mass forming there, just like the short-germ band insect. But then what it does is, boom, it makes all the segments not quite at the same time. There's some wiggling around, and then it emerges with a bunch of segments right there. This is what flies do. They make all these segments basically with some minor variation all at once. They're using similar molecules to do this. They're also using some different molecules. Fly segmentation is a complicated mess with all kinds of genes working together to generate it. Are they homologous? You end up with the same pattern. You end up with a segmented larva. So, if you just look at the outcome, you'd say yes, but the process is very different. And then you ask, why is the process different? It's because flies have undergone a period of accelerated development, that they've sort of compressed, squished down that complicated linear sequential pattern into just getting it done all at once, because flies have been selected for very rapid development. We see similar things all over the place in that, for instance, the quantity of maternal components affects the shape and development of an embryo. So this stuff has been around for ages and ages and ages. We call it the problem of homology. By problem of homology, we mean sometimes it's really tricky to infer homology. It does not mean, oh gosh, this huge problem of homology means that evolution didn't happen, which is how Wells and Nelson interpret it. So let me see if we can bring this back up. I actually looked something up. There's a really nice summary of the problem of homology in Mary Jane West Eberhard's book on developmental plasticity. So I'm just going to give you this brief paragraph because it really sums it all up and is our answer to the problem of homology. She says, changing characters do not march ever outward along the branches of a phylogenetic tree. While homology, parallelism, and convergence remain useful conceptual guides, they need to be seen against a background of continual reshuffling with a particulate mosaic phenotype that renders linear terms like parallelism and convergence only approximate and potentially misleading descriptions of evolution. So right there's the problem of homology, is that if you try to force things into this linear pattern, instead of recognizing the branching, divergent properties of evolution, you will have a poor fit with evolutionary theory. Does a concept of mixed or partial homology just make a mess of homology? In fact, evolution makes a mess of homology. So that's her summary, and that's a pretty good one, because basically what Wells and Nelson have done is they've turned the problem upside down. That what they have argued is that the problem of homology is a problem for evolution. And what everyone else is saying is, no, evolution's not a problem. The problem is that attempting to draw these linear pathways of evolutionary history via homology means homology is a problem. See, you see the distinction? It's an important distinction that Wells and Nelson are interpreting as a problem for evolution. Everyone else who's doing serious study in this field, unlike Wells and Nelson, are saying, no, evolution is not a problem, but homology is tricky and complicated. And that's the problem of homology. So I will leave you there. I hope that's enough to think about for a little while. And when I get back next week, maybe we'll have a session where we can have a little discussion about this, a little Q&A about the problem of homology, so-called, that Wells and Nelson have totally mangled, shamefully and intentionally, I'm sure. As I mentioned, Hovind and Ham, they're jokes. They're really not very bright, and they don't know much. So when they screw up, it's because just pure ignorance is at work. So this is the problem with Wells and Nelson, is that they have these ideas that are dishonestly presented. That, when I talk about people like Hovind and Ham, they're just pure ignorance. They just don't know anything, and they're winging it, making it up on the fly. That makes them kind of foolish and fairly easy to argue with, because you can just stump them with the evidence. Wells and Nelson are doing something rather different, in that they actually have a little bit of background knowledge. They've gone looking intentionally for things they can twist and distort, and then they maliciously, malevolently change the intent of the authors of scientific papers to present a false picture of dissent in the scientific community. So that makes them a little trickier to handle. It means you gotta know a little bit about Evo-Devo and evolutionary biology in order to handle their crap, and that's what makes this paper dangerous. Because people like my lay correspondent, who clearly knows absolutely nothing about this subject, are easily bamboozled by the Wells and Nelson approach. And even those of us who know something about the field have to stop and think and remember our citations. And, you know, when they say that, when they cite Perry Albert and Rudy Raff as supporting their anti-evolutionary conclusions, we have to think for a minute, wait, what did Perry Albert and Rudy Raff say? And we have to file through our mental notepad of stuff we've learned from those guys, those very smart guys, and we have to realize, no, they're just making this up. They're distorting what they said. So anyway, watch out for Wells and Nelson. Truly dishonest people. They're just making it up as they go along and taking little nuggets of truth and turn them into garbage. Okay, that's it for now. We'll talk to you later.